Morning, students. Today we discussed about plant hormone brassinosteroids, biosynthesis, metabolism, and transport of brassinosteroids. It's the third part of brassinosteroids chapter. Like gibberellin and abscisic acid, brassinosteroids are synthesized as a branch of the terpenoid pathway, starting with the polymerization of 2-farnesite diphosphate to form C30 triterpene squalene. Squalene then undergoes a series of ring closure to form the pentacyclic triterpenoid precursor of cycloartenol. All steroids in plants are derived from cycloartenol by a series of oxidation reactions and other modifications. Our knowledge of the biosynthetic pathway is the result of a combination of genetic and biochemical analysis. For the biochemical studies, periwinkle, Catharanthus roseus, cell cultures were used as they produce brassinosteroids in relatively high amounts. Radio-labeled brassinosteroids intermediates were used in feeding experiments and their metabolic derivatives were identified by gas chromatography, mass spectroscopy analysis. Coupling this type of analysis to genetic studies with brassinosteroid deficient mutants in Arabidopsis, tomato, and other species has allowed the identification of the complete biosynthetic pathways. This section presents these following. Here, campestrol, simplified pathway for brassinoloid biosynthesis and catabolism. The precursor of brassinoloid biosynthesis is campestrol. Black arrows here in the diagram represent the sequence of biosynthetic events, and solid arrows indicate single reactions, and dashed arrows represent multiple reactions as shown in catasterone, the immediate precursor of brassinoloid, can be shown synthesized from 2 parallel pathways, the early and the late C6 oxidation pathways. In the early C6 oxidation pathway, oxidation of 6 carbon of the B ring occurs before the addition of vicinal hydroxyls at C22 and C23 of the side chain. In the late C6 oxidation pathway, C6 is oxidized after the introduction of hydroxyls at the side chain and C2 of the A ring. Brassinoloid is synthesized from campestrol, differing by their size. C24 alkyl substitutions. Brassinosteroids are synthesized from campestrol, cytosterol, chlorosterol. While campestrol and cytosterol are abundant in plant membranes, cholesterol is present at relatively low levels. All three sterols are metabolized to a large number of intermediates in plant cells, but only a few of these metabolites have biological activity. We will illustrate a simplified version of the brassinosteroid biosynthetic pathway starting with the sterol progenitor campestrol, which is ultimately derived from cycloitinol, first converted to then prospective uses of brassinosteroids. Brassinosteroids were discovered as a class of growth promoting hormones, and researchers immediately recognized their potential application to agriculture. For the past 20 years, numerous small scale studies have been conducted to the test the ability of brassinosteroids to increase yields of crop plant. Brassinoloid has been found to increase bean crop yield based on the weight of seeds per plant by about 45% and to enhance the leaf weight of various lettuce varieties by 25%. Similar increase in the yield of rice, barley, wheat, and lentils have been observed. Brassinoloid also promoted potato tuber growth and increased its resistance to infections. Tomato fruit set was also enhanced by brassinoloid. In addition to such small scale studies, large scale field trials using brassinosteroid derivatives have now been conducted in Japan, China, Korea, and Russia. The results of the field trial have been highly variable and appear to reflect the degree of stress under which the crop was grown. A crop grown under optimal conditions shows little effect of applied brassinosteroids, while a crop grown under conditions of stress shows dramatic effects of brassinosteroids application on yield. Thus, it appears that brassinosteroids application is most beneficial to growth under stress conditions. Brassinosteroids have also proven to be a useful aid for plant propagation. Pre-treatment of woody cuttings of plant such as Norway spruce and apple trees with brassinosteroids enhance the rooting response. Micropropagation of cassava and pineapple by tissue culture has been improved by brassinosteroid treatment. The expression of a maize dwarf gene in transgenic rice increased grain yield, reduced brassinosteroid function can contribute to agriculture as well. For example, decreased brassinosteroid synthesis or signal in rice results in dwarfed plants with an erect leaf habit, which allows higher planting densities, leading to increased biomass and final seed yields. As researchers continue to explore brassinosteroids' effects on plant development, additional applications of brassinosteroids to agriculture are bound to emerge. So it's finished, and it's third part of brassinosteroid chapter, and we complete here biosynthesis of brassinosteroids and the use of brassinosteroids in agriculture field. So thank you, students.